Hello, everyone, and welcome. Thank you for coming. I am Kevin McCarl, and we'll be doing a webinar today called Mass Analysis of Oligonucleotides using ProMass. All right, so let's go ahead and get started. For those of you who don't know, which is probably none of you, I think all of you are oligo customers of ours, but we're Novatia. We're a small contract research organization just north of Philly. We've got clients in the chemical, pharmaceutical, and life sciences industries, and you may have heard of ProMass. Hopefully you've heard of ProMass before. That is the software that we're using on a daily basis to analyze your oligonucleotides and our oligonucleotides. And that's something that Mark came up with here, and we use every day, all the time. So I'll get into details about that. But that's coupled with our Oligo HTCS platform, which is the whole instrument software combo that we use to run these oligos. Just as a quick outline, we're going to start talking about ProMass, just kind of an overview and how we use that for ESI mass confirmation. Then I'll talk about that Oligo HTCS system that we use. Firstly, I'll talk about our high throughput analysis and how ProMass factors in with that. We'll go over the ProMass report and all of that detail. And then we can move on to the detailed impurity profiling analysis that we do for oligos. And finally, we'll wrap up with some discussion about the high-res accurate mass analysis that we do for oligos. So this is ProMass. This is the software that we use for deconvolution. This is what it looks like. So it's fully automated, and it does deconvolution and reporting for ESI LCMS data. The driving force behind ProMass is the ZNova algorithm, and this is what we like to call an artifact-free algorithm. So we get deconvoluted mass spectra without artifacts in them. And ZNova also comes with a lot of spectrum enhancement features, such as background subtraction, smoothing, and baseline correction. So this allows us to get nice, clean spectra for good, reliable data every time. And then once the data is actually processed, ProMass is capable of doing the data reporting as well. So that's got a lot of nice features. It basically spits out a report in an HTML format, so it opens right in your internet browser. And I'm sure a lot of you have seen this as customers of ours. We'll send you a link to our data portal, and you can access that HTML report. That's the same thing that we use in-house. So it's all, you see what we see. It's got the capability to search for multiple target masses, color coding for kind of quick identification. You can identify sequence fragments and also estimate purity. It does automatic purity calculations. And I also sort of hinted at ProMass HR, which kind of adds another algorithm, PPL respect algorithm, and that allows us to do deisotoping for exact mass determination. And I'll talk about that at the end. So a little bit more about ZNova. This is a component deconvolution algorithm. So basically it tests and determines the charge of every peak in the raw spectrum. So you can see in the bottom right-hand corner there, I've got a raw ESI mass spectrum, and there's some sort of blue trails leading up from each individual M over Z value. And you can see right next to those, there's a charge state assigned to each, the negative 18, 19, 20, and so on. That's the charge state that ProMass has assigned to that particular charge series. So ZNova is an intensity-based scoring algorithm. So it gives each peak in the raw spectrum a score based on how it relates to the other charge states in that series. And it basically only picks the highest scoring charge states and uses those to produce the deconvoluted output. And that is how we can minimize artifact peaks. It uses the multiplicity of the charge states as opposed to isotopic spacing, So we don't need high-res data to do this. And as I mentioned, we have signal processing like smoothing and background subtraction to help process noisy data, for example. So all of this essentially is just to say that ZNova and ProMass allows us to do clean deconvolutions for reliable confirmation of target components. So just to give you sort of a more grounded illustration of what we're doing here, I've got a little picture of a nucleotide base group here, and you'll note that the phosphates are deprotonated here. So that's going to be the case at pretty much any pH above 0, so most of the time. And any of these bases on your oligo could be holding a negative charge. So when you have, for example, a 60 base oligo, you could have that many negative charges. And you can imagine that could make for a potentially complex spectrum. So in the bottom left-hand corner, you have an example of a negative ion raw spectrum for a 20-former oligo. You've also probably all seen this. It's what we use as our oligo standard for our analysis. So we take all those negative charges and M over Z values in the raw spectrum. ProMass converts them into the spectrum in the bottom right. You see it's nice and clean, 3 peaks. We have our main oligo species is that tall peak on the right, and then 2 expected impurities in the bottom left. So all of that nonsense gets kind of cleanly replaced by the deconvoluted spectrum. So we have quite a wide range of applications that we can use ProMass for, especially coupled with our Oligo HTCS system and the different analysis capabilities we have. So we regularly do things, high throughput molecular weight determination. A lot of our customers send samples to us every day for that. And we also have detailed LCMS and even MSMS structural confirmation. We can do analysis and determination of challenging samples, such as very long oligos and also heavily modified oligos. Hello, by the way, to the people that are just coming in. Yeah, so to continue, when we're doing LCMS profiling, that can be really helpful for a lot of different things. You can get the target mass confirmation that you'd normally get with just the high throughput, but you can also look at things like sequence failure list if we have the oligo sequence. You can look at all of the different masses under each chromatographic peak. So if you're doing a separation, you get all these different peaks. Your mass spectrum may contain multiple different species, so we can look at each of those. We can also calculate peak areas and percentages for each of those chromatographic peaks and also the mass spectral peaks. And we can also look at things like metabolites, and we can even identify those metabolites with the right analysis. And then I'll talk just briefly at the end about how we can use MSMS data to help with oligo sequence confirmation. And as I mentioned, all of these results can be viewed from a web browser because it's all HTML. So that allows it to be easily shared. You can just send a link like we do to most of you guys. And you can also download results as a PDF or an Excel file all through ProMass. So that's what we do all the time. It's really easy to share stuff. So this is kind of a diagram for the general workflow of how we do oligo analysis here. So you can see in the bottom right-hand corner, again, I've got that oligo with the deprotonated phosphate backbone. So of course, we're going to be doing negative ion ESI. And we're going to get multiply charged ions most of the time. So we need the ProMass software to convert that raw multiply charged spectrum into the neutral masses that everybody is looking for. What we do is prep the samples. Typically, this is pretty simple for oligos. In a lot of cases, we can just dilute them to the concentration we need in water. A lot of times it doesn't get more complex than that. Occasionally we'll have to use a different solvent or maybe a slightly modified method to accommodate certain modifications on the oligo, but it's not particularly common. And then the buffers we use are just mildly basic, and that helps to promote more negative ion formation, which gives us better looking spectra, easier data to deal with. And as you might imagine, the phosphate backbone being negatively charged would have a high affinity for cations. So sodium adducts and potassium adducts are extremely common. These can come from things like glassware, for example. It's really hard to get rid of them, so that's why we do desalting on our system. So start with the sample prep, and then once the samples are actually on the instrument, they are desalted in line. And then after that, there's kind of a fork in the road. So I'm looking at the top diagram here again. We can either just go straight across to the right, go from desalting over to just performing the negative ion electrospray, and then we do the deconvolution and reporting. Or after the desalting, we can do the LC separation prior to doing the electrospray, and then we'll do the deconvolution and reporting. And just a quick note that we do the same basic thing for DNA or RNA. It's all compatible with both. So this is what the system looks like that we're using in-house, at least one of them in the top right. So this is our Oligo HTCS system, and this one has the Thermo LTQ-XL. That's the mass spec we're using. It's on the right-hand side of that image. And we have the Vanquish, which is the Thermo system for the LC. That's kind of all the other stuff on the left. The black and gray boxes, all of that is encompassing the Vanquish system. And that's got the 2 column compartments on the left, which are very important for our analysis. I'll go into detail with those on the next slide. We also have another version of this system that is, we call it the twin system. It's got a twin autosampler on it, twin PAL autosampler. And with that, we're using the Thermo Ultimate 3000. You may be familiar with that LC. And that basically allows us to get double the throughput. So you can see under the capabilities, if you're running just plate after plate of these high throughput samples that we often do, you can get about 1,500 samples a day with the single injection method. And again, that's what's shown in the picture here. With the twin PAL, you can do double that because it basically just adds on twice as many of everything, and that allows the auto sampler to kind of do dueling banjos and inject twice as frequently. And like I mentioned in our workflow diagram, we have online desalting, so that gets rid of all of those adducts. Unless it's extremely salty, then sometimes it still can't quite handle it, but we have methods to deal with those. So this allows us to confirm target masses and sequence failures, other impurities, pretty good mass accuracy here, usually within about a Dalton, depending on the size of the oligo. And then it all comes out in the nice HTML data report. Oops, excuse me. We also have PDF and Excel format. And just to go over some applications real quick, a lot of these I already mentioned, but DNA and RNA, therapeutics and diagnostics. You can do oligo QC, impurity determination, long oligos, and oligos with labile chemistries. So all kinds of different things that we can run on this. So this is a flow diagram at the top. And you can see this sort of parallels the image in the previous screen. So the black boxes that were the Vanquish are basically the left-hand 3 rectangles, and then the actual mass spec is the far right rectangle, to use the technical term. So you can follow the different flow paths here, the blue and the green. Hopefully you're not colorblind. I apologize if this is hard to see. Basically, the left pump and right pump are each doing separate things on the flow path. So the left pump is the loading and washing. So this pretty much injects the sample onto the oligo traps, which are in the left-hand column compartment. You can see they're labeled 1 and 2. So an oligo sample gets loaded onto that trap. And in the meantime, the right-hand side is washing, or excuse me, not washing. It is eluting. So while one oligo is being trapped on the left-hand trap, let's say, number 1, the right-hand trap would be eluting the previous sample. And at the start of each run, the 2 valves toggle, the 2 valves at the top and bottom of that left-hand column compartment toggle, and that basically just switches which oligo trap is being washed and which one is being eluted. So this allows us to reduce our cycle time We don't have to wait and re-equilibrate because while one sample is running, the other column is being essentially re-equilibrated. It's isocratic, so it's not a gradient, but it's just rinsing off the column to prepare for the next sample. And all told, we're able to do a single sample in about 40 seconds with our single PAL or our single injector method. And again, the twin will kind of double all of that stuff so that if we're running on a twin, we can do a sample about every 25 seconds. And then just to give you a little bit more info about that, we've basically got two pumps, as I said. One of them is running our weak buffer, and one of them is running one is running our weak and strong buffers in different proportions, and then the other is running our organic buffer, and that's basically the left solvents, the weak and strong buffers, are used to load and wash the trap columns, and then the right pump with the organic buffer is used to actually deliver the eluent and the sample to the mass spec. And you can see that I've got the scan parameters for the actual mass spec in the bottom left here. So we're scanning from M/Z 500 to 2000. This usually gives us a pretty good coverage of the entire spectrum that we need to see for these oligos. And we're also using 5 microscans with a max inject time of 50 milliseconds. And this usually gives us good signal to noise, but also keeps the scan time short enough that we can get enough data points that you would be sampling a typical chromatographic peak. And just quick note at the bottom, we optimize this with a flow rate of 200, in the range of 200 to 500 microliters a minute, which accommodates our right pump gradient eluent flow rate. So if you're curious what the actual data looks like, this is an example of 120 more oligo that we ran using the high throughput method. So the top left corner, there's kind of two spectra on top of each other. There's the LCMS profile, which is just the TIC response. And then just below that is the raw ESI spectrum for that TIC for this run. And you can see all told it's about 15 seconds. Goes from 0.08 minutes to 0.26 minutes. And then ProMass transforms that raw spectrum. You can see it ends up in a report at the bottom. That's kind of what the top level report looks like. And it converts that into the deconvoluted spectrum, which you see on the right. So all of that, complex, kind of hairy looking spectrum on the left becomes that relatively clean, tall peak on the right. And you get to look at it in this nice simple report at the bottom. You've got the data file name, the oligo sequence that we typed in there with the target mass and the observed mass, and then intensity and purity. So all of that is nicely wrapped up for you. You can see it's green because it matches quite well. If, for example, we were running on the twin system, we might be running plates like these. So a 96 well plate might look something like the top image, and a 384 well plate might look something like the bottom image. And I just put the little color code key here that we have. So essentially, this is a quick way, if you're running all of these dozens or hundreds of samples, to just kind of eyeball and say, okay, which ones are problem samples and which ones are good to go. So this gives you an easier time of poring over your data, not having to necessarily check every single sample in detail. So just to get into a little bit of the report itself for each sample, if you actually click on any of the, I'll just go back real quick. At the bottom of this, you can see all of these labels are actually hyperlinks. So if you click on any of these at the top level report, it'll take you to this kind of expanded detailed report. So this is still for that 120 mar, but you can see all this extra detail on the top left, the injection volume and position. We've got the whole oligo sequence is now visible. And then, we've got all of these different reported items, the target mass, the observed mass, the error between the two, which in this case is 0 Daltons, pretty good. And then we also have abundance in spectrum, which is related to the mass spectral abundance, and the purity estimate, which is the product of the mass spectral abundance and the chromatographic abundance. So this can give us just a quick view, but with a little more detail about how successful our actual run was. And again, you can easily share this data. It's not some proprietary software that you have to use to look at the report. You can have ProMass and share this with a friend of yours that doesn't have it, no problem. A little more detail on the report. If you scroll down, you actually get into the chromatograms and spectra. So on the left-hand side, I've got a zoomed-in version of the deconvoluted spectra. That's just zooming in to a range of like 150 Daltons around the target mass peak, or the base peak. And then on the right-hand side, it's a little preview of our interactive spectrum viewer. And I'll show you that with a little live demo in a minute here. But basically, the top spectrum is the deconvoluted spectrum, and you can see there's kind of a miniaturized version of that just below. And that allows you to actually zoom in on the deconvoluted spectrum wherever you'd like, look at different regions. And then you can see the bottom section is the raw spectrum, and that has all of those blue callouts like you saw at the beginning of the presentation. And so this basically comes from clicking on a peak in the deconvoluted spectrum. This will automatically label all of the charge states that were assigned by ProMass for that peak in the raw spectrum. And just like the deconvoluted one, there's a mini one below you can use to zoom. I'll show you how to actually interact with this in real time, but this is kind of a taste of how you can really dig deep into our reports. And then we also have a deconvolution peak report. So if you put in an actual oligo sequence, this can call out different mass differences that it notices. For example, it can call it depurinations or depurimidinations. It can call it sequence fragments. It can call it adducts. I've highlighted just a couple of these for this 120mer here. On the right-hand side, you've got the target mass labeled, and then it actually tells you, since we entered the oligo sequence, which bases that starts and ends with. And then just below that, it's saying, okay, this is either a T depurimidination or an A depurination, and that's just because the mass is close enough to both that it could be either one, but it helps you to kind of figure out what you're looking at. And then sort of on a similar train here, we can also look at modified oligos. So if you put in the oligo sequence, you can just type it into Excalibur or MassLynx or whatever software you're using, and ProMass can actually read it and calculate the mass automatically. So in addition to just regular plain old bases, you can also put in modified bases or even custom modifications. There will regularly be oligo modifications for samples that we receive on the 5' or 3' end, sometimes even internal modifications. So we basically have a text file that ProMass references that uses input numbers to calculate the masses of all of these different things, so that you can just type in the sequence and then calculate the mass. So just for example, on the left-hand side, this is what a little snippet of the text file looks like. You can see the bottom left-hand corner, I have phosphorothioated RNA. So we would just put an RA star in the sequence to represent that base, and so on. And then on the right-hand side, kind of the larger image there, this is an example of what you would put in for a 5' biotin modification. So you would just put in that little /5 bio/ in your oligo sequence, wherever that would be on the 5' end. And then ProMass would actually calculate that. And then, when it comes out in a report like this, you would actually see that listed out in the presumed identity when the mass matches. So you can do any custom modification that you like as long as you define it in that same format. You basically just have to take the mass of the monomer plus a linkage phosphate and minus the mass of water. And that's the mass that you would put into this reference file, and ProMass can calculate it for you. So I'm going to show you a live demo in a second here of kind of just how a typical ProMass processing event would occur. But these are basically the 5 steps to actually get from your data input to your results output. So I'm using Excalibur, but you may be using MassLinks, or you may be on SciX or Shimadzu, for example. ProMass works with all of those. But basically, just for the sake of Excalibur, to make it easy, I'll go through that. You just need the sample ID, processing method, the ZNova parameters, which are what kind of tells ProMass what to do, and then target info. So once you have all that information, you enter it into Excalibur, and then ProMass is actually layered on top of Excalibur. So you put all of that info in Excalibur and acquire your data like you normally would without ProMass. And then there's an Excalibur peak picking method that you would use. So in the bottom left-hand corner, you can see kind of what that looks like. It takes the spectrum and highlights different regions, and you can sort of tweak which peaks are picked and not. And then, so that's all kind of on the Excalibur end. And then once all of that's done, once the peaks are picked and your data's acquired, it sends it off to ProMass. And that's where step 4 comes in. So this is kind of what it looks like to be building the ProMass parameter file. You've got all of these different things that you can tweak and change to improve your ProMass results. And then, so Excalibur will do all of its peak picking and such, pass that data on to ProMass. ProMass will deconvolute the spectrum and then give you that nice clean HTML output. So just bear with me 1 second. I'm going to switch over to my virtual machine so I can share that screen, and I'll show you sort of a live demo of how that actually works. So hopefully you can all see my Windows VM now. I've just got kind of a folder open here. The data is already acquired. It's a set of various oligos at different lengths. So I'm just going to open this Excalibur sequence. And so again, this is in Excalibur, and these are just all of the samples for the data I've already acquired. I'm just going to go ahead and reprocess these. So this is within Excalibur. I can actually set it up to use ProMass. So it's already set up to do that. I just have to click OK, and it's going to pass that along to ProMass. So you can see it's kind of processing right now. And in just a few moments here, we'll have all of these samples in a nice ProMass report for us to look at. So I'll just open ProMass here and click on this ProMass browser button. So you can see it's all done. I just processed, like 8 samples. You can see we've got the sequence info, all the target masses, observed masses, purities, all that stuff that I talked about. I'm just going to open a report here. So again, you just click on any of those hyperlinked items and it opens a more detailed report. So we've got, in this case, I'm looking at a 6-dimer, and you can see the nucleotide sequence is right here. And I get all of this nice information just below that for the abundance, which in this case matches the purity perfectly because the LCMS area is 100%. There's just the one peak because we're just running essentially a flow inject. It's just the high throughput method. And then it's got the full sequence listed out here because that's what we found, base C1 through T60. And if I scroll down, I can see this is the TIC and the LCUV. And then I can show you the raw spectrum and the deconvoluted spectrum. And then I just want to show you the data viewer kind of in motion to show you what it looks like. So again, I can sort of, I'll just show you the raw spectrum before I click on anything. That's the raw spectrum. And then if I just go ahead and click on the deconvoluted mass and then go back down, you can see it's highlighted all of those charge states again. And then I can actually zoom in by clicking on a certain region and dragging. If I want to say verify those charge states, looks like they're all matching up quite nicely. So you can share those results and come back, open any of the other reports There's a lot of versatility here. Okay, so that was all essentially related to the high throughput, and now we're going to take that top path in the sample workflow that I've highlighted in red here for the LC separation. This is what we would do if we're doing impurity profiling. So you can see this is a very similar flow diagram to the previous one. The main difference is that this time the second from the left rectangle there has an analytical column in it, and this time you can see it's in the elution path. So that basically means that we're actually putting the sample onto the analytical column for separation. We're still desalting. You can see it passes through the oligo trap. That's still a very important step of the analysis. It's just that it does the desalting and then it gets separated on the analytical column after that. So essentially this is going to be ramping up the gradient, and we're going to wash the column off and then re-equilibrate. And we have a couple different versions of methods that we run at Novatia, 5-minute and a 20-minute gradient. Those are kind of our canned methods. You're obviously able to modify those methods if necessary, but those typically get the job done. And just a quick note: this system is also UPLC compatible. So if you want high-res applications, chromatographic resolution, I mean, you can do that. Or if you want to maybe shorten the method time up, that's also a possibility. So these are the method parameters. We're using the same buffers actually, but we're just adding an additional buffer on, or it's more of a wash buffer, the 40-40-20 methanol, acetonitrile, and water, and that's just used to kind of rinse off the column at the end. So if you look at the flow diagrams, or excuse me, not the flow diagrams, the gradient diagrams at the bottom, the green block on the right-hand side, let's look at the right pump. That kind of represents the gradient ramping up, and then the blue rectangle you see is the wash. So for this method, either the 5-minute or the 20-minute method, we're using a 2x50mm core shell C18 column, and the flow rate is 0.4 mils a minute, and we're running the column oven at 60 degrees C. So this, I guess I didn't clarify this. These gradient that you're seeing are both for our 5-minute methods. It's essentially the same shape for the 20-minute, but obviously it's spread out over more time. So the gradient itself is 4.5 minutes, or I guess it is 5 in this case. I put the wrong time up, I apologize. But the total run time ends up being 9 minutes for that. So this is an example of some samples that were run using, or excuse me, one sample mixture that was run using this method. So this is one mixture of 7 oligos on our 5-minute LCMS method, and you can see they've all separated fairly well. The 20 and 21 mer start to kind of overlap a little bit, and then the 29 mer and 30 mer are definitely co-eluting, but you can also see a clear separation between the two. This is just sort of to illustrate that we can effectively separate, you know, a mixture of all these different oligos that are, in some cases, only one base apart in length from each other. And it works out pretty well. And then ProMass comes in, and we can actually look at all of those separately in the same report. So we've got, you can see at the top, 7 different target masses, and they were all found and all reported in the ProMass report. I've gone ahead and highlighted the purity estimate for each of those too. Basically that, again, factors in the mass spectral abundance for each chromatographic peak multiplied by the total chromatographic purity. And that's how you end up with those purity estimates there. And then below that target mass summary, we also have the chromatogram summary. So that's the really large table with an almost overwhelming amount of data on it. So I stuck some labels on the right-hand side to make it easier to pick out the different oligos that are in there. So, you know, the 6 mer all the way down to the 30 mer are each highlighted. And, you know, there's all kinds of different impurities and additional sequence fragments in there that we're seeing. These days, oligo mixtures are very common targets. For example, probe mixtures, we get those all the time. So this type of method is really good for taking a look at all of those different things in a single mixture. Oh yeah, and just to mention, if adducts are present in the spectrum, ProMass will actually not, or it will account for those by removing them from the purity calculation. So it'll tell you in the peak report how much of an adduct there was, but it won't actually factor that into the purity because it's coming from the mass spec analysis as opposed to being present in the sample itself. Yeah, to sort of expand on that a little bit further, this is an example of a metabolite ID study. So you can see we enter the oligo sequence in, and on the right-hand side of the giant table, we've got all of these fragment sequences coming up in the sequence ladder summary. So again, that tells you that there's partial sequences coming out, and it's actually able to identify those based on the target masses. So in this case, we found over 40 metabolites, and they were all automatically identified. Really saves a lot of time. You don't have to go through and pick out, calculate each mass, and think, what could this be? ProMass just does it for you. Something to note, however, this was actually high-res data as opposed to average mass. All the data we've looked at up to this point was average mass. This is high-res. So this is kind of part of what allows us to get more confidence in these impurity identifications, the metabolite identifications, rather. So all of this has mass accuracy within 3.3 ppm. In a lot of cases like these, the biological matrix can contribute to false identifications if you're just doing average mass. So the fact that we're doing monoisotopic mass determination is very important. So to sort of expand on that, on the left-hand side, I've got some information for an average mass determination of a 6-dimer, and then the right-hand side is the monoisotopic mass determination of the 6-dimer. So the left hand was measured on an LTQXL, and the reported mass was 18078.6 versus the target. It was off by 1.2 Daltons, which on a ppm scale amounts to 66.3 ppm. So that's quite a lot. For an average mass determination, 1.2 Daltons isn't that bad, but if you're really trying to unequivocally identify something, that's not exactly close. Contrast that with the right-hand side, which was actually measured on an orbit trap. You can see we've got several more decimal points, which helps quite a lot. The mass error for that was only 0.22 ppm. So as long as we have the isotopic resolution to get the high-res data, we can use ProMass to actually de-isotope and give us that very minimal mass error for the high confidence identification. So with that, I'm going to move on to the high-res stuff. So ProMass HR is kind of ProMass Plus, if you will. It's got all of the ProMass Znova stuff, but it also includes an algorithm called PPL Respect, and that allows us to do de-isotoping. So you can see in the bottom right-hand corner, that is actually a demonstration of how PPL does peak shape modeling. So it actually reconstructs the data without noise, and it does that before applying charge deconvolution or the de-isotoping algorithm. And so that allows you to get a good clean representation of the data that kind of is able to be applied over the entire spectrum. So this accounts for changes in the peak shape or width across the spectrum. And when we're doing de-isotoping, the mass accuracy essentially ends up matching the same mass accuracy of the instrument. So for example, with an orbit trap like we have, or QTOF, it's typically less than 5 ppm or up to 5 ppm. Whereas the typical mass accuracy for average mass with unresolved, isotopically unresolved data is more like 50 to 100 ppm. So that's quite a lot. And this allows you to get fewer false positives from chemical background, for example. In the case of oligos, there's another feature that's particularly important if you have modified oligos. So you can actually input a custom unit cell molecular formula to account for different ratios of atoms. And this makes the de-isotoping much more accurate. So for example, if you're working with phosphorothioated oligos that have sulfurs in them, If you don't account for those sulfurs when you do the deisotoping, you're going to get a lot of artifacts, deisotoping artifacts, from those extra sulfur isotopes that aren't being accounted for. But this here within PPL in ProMass HR allows you to account for those things and improve your deisotoping. In addition to this, if you don't actually have isotopic resolution, PPL and ProMass can actually work together to still improve the resolution of your data. So you can kind of clean up an average mass spectrum even if you don't have isotopic resolution. So that can also be really handy for just making your data look really nice and clean. So as an example of the data acquisition here, this is a 21 mer collected with high-res accurate mass, and the kind of expanded spectrum in the bottom right is showing the -8 charge state of this oligo, and I've labeled the monoisotopic peak on the left-hand side there. And basically, we're just taking all of this, you can see that's got very good isotopic resolution, and that's occurring for every charge state across the spectrum. So we're able to take this very complex spectrum and make it into just a clean single line with the final mass here. And just to give you a little bit more of an example of that, I've highlighted the 21 mer in this table just so you can see that's the one we were just looking at. But we've got all these other oligos in here too, 6 mer up to an 80 mer. And in most cases, the mass error is actually under 2 ppm. You can see at the 67 mer, it's a little bit higher at almost 3 ppm. But for the most part, it's very, very good. You'll notice that the 80 mer was actually not determined, and that's because we basically reached the limit for isotopic resolution. So these were all at 30,000 resolution on the instrument, and once you reach a certain size, your isotopic resolution starts to go down, and you essentially just can't pick out any actual data. So you can't determine the mass of the species you're looking at. So just as sort of an illustration of that, you can see this is a 67 mer. In the top right corner, I've just got sort of a zoomed in spectrum, and you can see there are a couple of artifacts around the main peak there. So this is sort of just starting to push the limits of the deisotoping here. And the bottom spectrum, you can see there, the monoisotopic peak actually looks like it's essentially being extrapolated by the data at this point. It's so small compared to the rest of the data and the isotope pattern that it's almost invisible. So as you get more and more charges, the distance between the charge states narrows, and you need more resolution to actually be able to use that data. So essentially, we're limited by the resolution of the instrument and also the increasingly low intensity of the monoisotopic peak as you increase in mass. So just the last thing I want to touch on is the potentiality to use ProMass HR for oligo sequencing. It's not fully automated, but it is an important asset that can contribute to oligo sequencing, and we're working toward making it more automated. So essentially the steps for the sequence confirmation are to get the intact mass of the oligo by running negative ion ESI. We're getting high-res data, picking a low charge state for MS2, typically somewhere in the -4 to -6 charge range, then getting the MSMS product ion spectrum, and then we can use PPL respect to deisotope that spectrum and get a simplified fragment spectrum with all of our exact masses. And then we can basically compare that output, that simplified product spectrum, to the predicted fragments and compare the two to get our oligo sequence. So the tail end of that is definitely a lot of manual work right now, but we're working toward getting this to be a much more simplified and automated process. So in summary, ProMass Standard and ProMass HR can be used to provide rich ESI data and information on both standard and high-resolution instruments. We can get quick and reliable molecular weight determinations on our high throughput system. We can profile impurities and degradants using LCMS, and in addition to that, we can predict and match the degradation products using the target sequence. And that report has all kinds of useful information in it that you can easily share with your colleagues or come back and reference anytime. We can use high-res accurate mass deisotoping to get monoisotopic mass for the most accurate method of identification from basically 0 to 20,000 Daltons, which is very applicable to therapeutic oligos and their impurities and metabolites. And then again, we're working on using this to help with things like oligo sequencing in the future. So with that, thank you all for attending. I appreciate your patience and attention. Please feel free to type any questions in the chat, or I think at this point we could probably also try some microphone questions. If anybody wants to let me know, I can unmute them. Otherwise, I'm planning on posting this recording up on our website at some point, so you can come back and watch it anytime. But yeah, thanks again, everyone, for watching, and I'll take any questions if anybody has them.
This is John. Thank you for your presentation.
Of
I had a
course.
question. I had a question regarding the reproducibility of basically the purity values that you're getting.
Okay.
Would this vary from day to day, or just from differences in, I don't know, buffers, temperatures, whatever? I'm just wondering how consistent
Sure.
some of these values are.
Yeah, so I don't have an exact numerical value to give to you for that, but I can say that the variability in purity is probably going to be pretty minimal. It should be fairly consistent. The main things that I would say contribute to that are going to be the amount of sample that's injected and the signal to noise that we can get from that. If it's a very low amount of sample, the purity variability can be a lot greater because you're going to see more noise interference. And sometimes if noise gets picked up and deconvoluted as real data falsely, then that could skew your purity one way or the other.
Okay, and I guess I potentially missed it for a second. You said that when calculating kind of your, at least your purity, that the adducts are accounted for, as long as I guess they're listed in your deconvolution peak report.
Yeah, so I probably wasn't as clear as I should have been there, but essentially, if ProMass finds an adduct, it will list it in the report and say, hey, I found this. But when it actually calculates the purity, it will not include the adduct in the calculation. It knows to exclude those adducts because it knows that they're not an actual part of the sample. Does that make sense?
Well, but if I just have some sodium adduct, it wouldn't I want to include that percentage? If I'm trying to, let's say, because I've been putting in internal standards and trying to get kind of a correlation to basically yield off of these little supports that we're using. And I'm wondering, since if I have a large adduct peak, and I'm trying to compare it accurately to this internal standard, whether or not I should include the peak area for that adduct or not.
Sure. So in most cases, well, this is obviously not applicable to everything, but we hope for a smaller adduct peak. If there's a really high adduct percentage, it might actually be more worth it to change the instrument method or the source conditions. And this is actually, maybe I could answer Daniel Fletcher's question in the chat here at the same time. He asked what can lead to a depurination during the analysis.
Mm-hmm.
So there's kind of a balance here between depurination and adduct formation when you're tuning the source for the ESI. So if you go in too hot, so to speak, you can get a lot of depurination, but if the source conditions are not strong enough, you'll have more adduct formation. So you can do some things with the ion source to sort of tweak that to balance between those two things. As far as reporting the adduct formation, I suppose it could come down to a personal choice. We typically tend not to report the adducts in the calculation for purity because, again, they're not really part of the sample so much as they are just coming from the mass spec process.
Okay, thank you.
Sure, thank you for your questions. Anybody else have any questions right now?
Okay, one more since no one else is jumping
All right,
in.
thanks, John.
Where do I type in the sequence? So if because typically I just give you masses, I don't actually give you the sequences involved.
Sure.
But can I basically include that sequel or type in that sequence once I
Yeah.
have a generated report?
So we would basically need to do it. I'm assuming you're talking about like because you're one of our oligo customers, you're not running ProMass on your end, right?
Correct.
So we would need to input the sequence on our end into
Gotcha.
Excalibur and reprocess the data. But
Okay.
if you're sending in samples, you're welcome to include the sequence. You can send a sequence instead of a mass or in addition to a mass. It's usually good to send both just in case. We've had probably countless times where people will send a miscalculated mass based on the sequence.
Gotcha. Perfect. Thank
Um,
you.
sure. Okay, well, it seems like nobody else has any questions. I'm going to slowly back away just in case I'm wrong. Thank you all again very much for attending. I appreciate your time and attention. I hope you learned something. Um, and yeah, I look forward to speaking with you all in the future. So have a good afternoon. I'm going to go ahead and end the meeting now. Oh yeah, feel free to email me with any questions if you have any after the meeting. And again, I'll try to post this online. So,
Thanks
all
very
right.
much, Kevin.
Yeah, of course. Thank you.